Ouais, moi aussi. Il est bien dans la classe, je sais pas, c'est pas. Non, mais je pense que le processus des enjeux
Oh, I just finished some everything now, it's a bit late. Yeah, but it's okay. It's a bit late. Les autres sont bons.
It's my former PhD student who made all the drawings.
Oh, yeah.
I'm actually not artist.
What did they use? It's like ink stain?
Yes.
Okay, Karen is queen as well. She's made all of her skills. But I'm no good at that.
Very jealous of them.
Juste parce qu'on n'a pas le cerveau que tout le monde a voté.
Hello everyone, welcome to the second in-person seminar of the year in the safe room. So as you see, I'm still here trying to do the technique, but hopefully I had two students who did a wonderful job today. So everything is working. So it's a webinar mode. People on Zoom, you cannot talk, but if you have questions, just ask on the Q&A. And at the end of the talk, I will open your microphone for you to ask your questions. And now I give the floor to Lila, who is going to introduce today's speaker.
Is this in front of the, I don't know where.
Yeah, but it's...
Okay. Hi, everyone. Thanks for coming. Sorry about all the confusion with the QR codes. So I'm really excited to introduce you to Kai Lucek today. So Kai is an assistant professor at the University of Neuchâtel. So where he now mainly works on butterflies. He started working on sticklebacks with Ulf Seehausen for a master's and a PhD in Bern. He then did a postdoc in Sheffield with SAP's own Patrick Nozzles, working on walking sticks. Then he did some botany in Basel, so he's kind of been in every study system. And now he's in Neuchâtel on an Excellenza fellowship, working mainly on butterflies, but as he said, he'll also work on any system that his students bring to him.
Yeah, thanks, Lila. Thanks for actually hosting me here today. So as you heard, I have a fishy past. Today I'm mainly talking about butterflies, alpine butterflies, talking about a macroevolutionary pattern and microevolutionary processes. Okay, we'll figure that out. So my talk has basically two parts. The first is more on the macroevolutionary impact of varying chromosome numbers and the genomic architecture, at least as far as we know it. And then the second part is more on secondary contact zones on primarily the butterflies. And I have to acknowledge, I have now many people in the group. They all have exciting projects. I don't have the time to show you everything that we do. And if you wonder why Kenny is here, Dylan is just so much in the lab, so I didn't have time to get a picture of him. But sometimes you will actually see people popping up, meaning it's actually their research that they present. Speciation. As you know, there's a lot of disagreement how we can actually come to a new species, how species evolve, etc. But there's also some agreement. So we can probably agree, all at least I hope, that for the advancement of speciation, we need barriers to gene flow to evolve. And there's different types of barriers that can be prezygotic, that can be postzygotic. When you think about postzygotic barriers, we often think about genetic incompatibilities. So if you have two parental species with a different chromosomal rearrangement and they mate, then you have a hybrid that's probably less fit or even sterile. And when we think about genetic incompatibilities, we often think about genetic rearrangements such as small ones, deletions, duplications, and especially inversions got a lot of attention for in the context of speciation. But today I'm actually scaling a bit up to something larger, namely chromosomal rearrangements that lead to chromosomal fusions and fissions here in the bottom right. Okay, getting better. And chromosomal speciation theory used to be a thing. So there's this classic type theory developed until the 1990s. And the idea there is that rearrangements, so different karyotypes, would actually lead to hybrid sterility. So as a consequence, differentially fixed rearrangement will quickly result in strong reproductive isolation. The problem here is, though, that if you have a rearrangement, it's really hard to establish or even become fixed. Because imagine you have a new rearrangement. If you mate with someone who doesn't have it, the offspring is probably being less fit or doesn't even occur. That's why more recent takes of this theory suggest that rearrangements can actually lead to reducing recombination locally in the genome and through time lead to speciation. In this case, rearranged chromosomes would eventually become fixed either by drift or through selection by, for example, fusing together chromosomes with different adaptive loci. Nevertheless, if you talk with people that work on chromosomal speciation is deemed to be unlikely. Why is this? Actually, I realized something when I looked at this theoretical approaches, namely that people thought about only about monocentric chromosomes. So this is chromosomes that we have with. Anyway, so it's chromosomes that we have with a centromere. And if you have, for example, a fission event, part of the chromosome gets detached. If that part doesn't attach to something else, it has a centromere, it gets lost. We can imagine we get an outcome that likely has a lower fitness than the parental stage. However, more than 10% of the extant eukaryotic diversity doesn't have monocentric chromosomes. They have so-called polycentric chromosomes. So holocentric chromosomes have small centromere-like structures spread across the genome. As a consequence, if you have rearrangements, the rearranged parts don't get lost. So we can imagine that we get actually something out that has a similar fitness as the parental stage. So why I'm talking about holocentricity? So these are two closely related butterflies, and butterflies have holocentric chromosomes. It's actually something I didn't know when I started working on them, but when I figured it out, I was actually quite mind blown. So these are closely related species. They have about the same genome size, but when we look at the karyotypes, they differ vastly. So on the left, we actually have the species with the highest karyotype count that we know of, with more than 200 chromosomes. The one to the right just has a few dozens. And it's only evolved through chromosomal fusions and fissions. So there's no genome duplication or polyploidization. And we see the same thing happen in other holocentric groups like Carex, Setches that we also work on. So we also have close related species that differ vastly in chromosomal numbers. And does this actually have an impact on speciation? So we suggested that we can actually use a phylogenetic model of trait evolution. So along phylogeny, we could imagine we have so-called cladogenetic events where you have just karyotype evolution at the speciation event. So you evolve a new chromosome, you split, you have a new species. Or we can also imagine we'd have anagenetic genetic events where karyotype evolution would evolve along a branch and eventually the species would split and establish new karyotypes. Both are of course not mutually exclusive and we could have contribution of anagenesis and cladogenesis. And what we suggested is that this reflects pretty well two types of chromosomal speciation models, so that cladogenesis is more like hybrid dysfunction model where you have rapid speciation, then anagenesis is more this type of combination suppression type models where you would slowly build up barriers and eventually speciate. And to assess the contribution of anagenesis and cladogenesis, we established that in our pet system, which are Arabia butterflies. So Arabia butterflies have more than 100 species. It's one of the most diverse groups of butterflies in the Northern Hemisphere. We have 27 species in Switzerland and some cryptic ones that we still try to investigate if we have actually more species. They're often called adapted, so that means alpine regions represent, like they also present biodiversity hotspots. And shown here is, for example, in the bottom right is the idealized distribution of the different species that we have along an elevation gradient. They're diversified over 15 million years in Europe, and some groups are actually much younger. And of course, they have varying chromosome numbers. So shown here are the different subgroups that we have in Arabia. We see there's a lot of variation between them. Some of them are relatively boring, just about 30 chromosomes, and others like the Tyndarus group, which is also the youngest and most species-rich group, they go crazy. They have between 7 and 51 chromosomes. And they actually also, it's the youngest group that only evolved over the last 2.5 million years. So what we did, this is now the work of my former PhD student, Hannah, who's now a postdoc at the University of Fribourg. So what we did is we obtained samples from more than 80 species. We went for whole genome resequencing. We extracted about 3,000 single copy ortholog genes, which we used to establish a dated phylogeny, and we ran chromoassessy. Chromoassessy is a model that allows to estimate contribution of clado and anagenesis for chromosomal fusions and fissions. And behold, this is already super exciting result. So this is the first almost fully resolved phylogeny for Arabia. If that isn't exciting enough, and hopefully this will excite you, this is the outcome of chromoassessy. So we can basically see the contribution for the rates of speciation for anagenesis, phylogenesis linked to fusions and fissions, respectively, and also without chromosomal change at all. So we see the rate of speciation is higher when we have chromosomal rearrangements involved. And indeed, most speciation events actually involve chromosomal rearrangements. And we also see that there's a difference between anagenesis and phylogenesis. So, fusions seem to be more often have an anagenetic component, when fission events result more often in cladogenesis. So we have here, along using macroevolutionary inferences, evidence that they actually do something different. So fusions and fission have a different impact. This is along the entire phylogeny. And we also looked at the different subclades for which we had most karyotype information to see, is there actually some variation going on? And running this, sure enough, we see that the contribution rates of speciation differs among the different subgroups for cladogenetic and genetic components, fusions and fissions. So basically it varies among clades. Nevertheless, just looking at the overall rate of speciation, we can see that the rate of speciation is actually highest in the clades that have the most rearrangements, suggesting that rearrangements drive diversification in this group. And this is something that we already showed before with just less data, but for more genera. So we have here the rate of chromosomal evolution against the rate of diversification. And what we show is that genera with more variable chromosomes diversify actually faster. To conclude on this, so we see that varying chromosome numbers actually have an impact on rates of speciation. The underlying mechanism, so that's fusion versus fission, anagenesis versus cladogenesis, seems to differ though among the different groups that we study. And chromosomal changes seem to be associated with bursts of diversification. We have this kind of link between fusions and fissions to speciation. We don't have, this is all correlational evidence so far, but what we're now interested in is actually trying to understand what is the underlying genomic architecture of chromosomal fusions and fissions. Because what I did not tell you so far is that not all butterflies go crazy with chromosome levels. Actually, most butterflies have 31 chromosomes. Only some groups do this, even within Arabia. So we try to understand what causes fusions and fissions. So we try to basically find the needle in the haystack. And to do that, we first generated our genomic haystack by sequencing and assembling 12 chromosome level genomes. So this is now work by my PhD student, Camille. We also got 3 genomes from the Darwin Tree of Life initiative. And then we also became part of this recently launched project called Psyche. So Psyche aims to sequence reference genomes for all European Lepidoptera, about 10,000 species in the next 5 years. So the first 500 is already online, another 500 by the end of the year. If anyone wants to join it, please discuss with me later on. And to give you an impression of our haystack, this is symphony. This is basically a symphony plot for a subset of the species that we have. And what we can see is there's way more rearrangements than you would expect. So here on top, you have 2 species that actually have the same karyotype. But we see they have actually way more rearrangements than you would have expected. So everything that I showed you before, it's just an underestimation of all the rearrangements that we could imagine, right? We also see there's more differences. And we look at different types of chromosomes. We actually realize there's interesting things going on with sex chromosomes. We have, for example, here a case where an ancestral W chromosome fused with an autosome, creating a second Z chromosome. We also have a case where you have a loss of the W chromosome. You will see that in a moment. So that means we have a large haystack of genomic data, more complication when it comes to sex chromosomes. And now we have to figure out what are actually the needles that we look for, right? We have some that we try to look for, including repetitive elements. So why repetitive elements? So including transposable elements, satellite DNA, and ribosomal DNA, because they can actually cause interchromosomal rearrangements or through ectopic recombination, so that you have clusters of the same repeat type recombining across chromosomes and therefore causing rearrangements. Looking at it so far, so what my PhD student Camille did, so she looked at breakpoint windows by comparing the sister species and looked at the distribution of repeat elements and compared it to random windows across the genome. And what we can see is basically the breakpoint average here in orange overlaps with the random distribution. So overall, we find no enrichment for different repeat classes for neither for fusions or fissions. But mind, this is at the class level, so now she is getting deeper in the annotation of the transposable elements. We cannot exclude that there could be something going on, so it stays exciting. We also look now more into the 3D genome structure, because when you work on genomic sequence data, you may think that the genome is just this linear sequence that you have, but of course, in the cell, it's folded in a very complex three-dimensional structure. Where you have different chromatin loops, chromatin compartments, or chromosome territories. And we just published an opinion piece in Trends in Genetics where we posed the question, is there a link to speciation? I don't have an answer yet. But we know that the 3D genome structure actually affects the gene expression, but also the recombination landscape. Similar chromosomal rearrangements can actually affect the three-dimensional genome structure. And what we suggest is that potentially this could also be linked to polygenic selection, because what is far away in the linear genome could be actually very close by in the three-dimensional structure. And what it looks like so far in our system, shown here is the three-dimensional reconstruction of our Arabia genome. So we have here on top the species with the lowest chromosome number, so 7, and the one with the highest chromosome number, so 51. And what we can see, if they split them up, we end up having small chromosomes that look like small balls. But when they fuse together, they still have like this ball structure. So we have like a necklace of pearls. And we try to understand now if this is actually linked to the synteny blocks that we observe. We don't have an answer to this yet. What we looked so far is at the contacts between different chromosomes. So using high-C data. So you see the darker it becomes, you have more contacts, and what we find, there's actually no difference between fused and non-fused chromosomes. Nevertheless, what we see is that smaller chromosomes have more interchromosomal contacts than larger ones. And the chromosome with the highest interchromosomal contacts, which is here, with the very dark spots, is actually always the conserved ancestrally smallest chromosome. So even though we have million years of evolution between the species, there seems to be part of the three-dimensional chromosome structure that is conserved. We also look now into methylation. So methylation, the idea is that it could regulate the expression of genes, but also mediate the expression of repeat elements. Here, we went also at the macroevolutionary scale. So we went for whole genome bisulfite sequencing for more than 40 species that we had in our freezer. What you can see is overall, we have relatively low level of methylation, just a few percent, which is actually what you would expect for Lepidoptera. We find there's differences between sexes, but there's some variation among the different families. Sample size is not that high. We actually get more reference genomes soon, so we will have more data on this. This is all just preliminary. So what we currently do is we annotate the repeat landscape, do the synteny analysis across all the species that we have, and try to identify the breakpoints and the associated methylation patterns. So we don't have the fine scale analysis yet, but at large, just looking at the repeat content of a genome against the percentage of, that is methylated, you see phylogenetically corrected, there's a positive correlation, suggesting that an increase of repeats could actually increase methylation as a regulatory response. And of course, you can see there's this one group here of nymphalids, which are outliers where the slope is much steeper, and those are Arabia. As usual, they do something else. Other things that we look at now are gene gains and losses. So far, we only have annotations using ab initio annotations. We just recently got our RNA-seq data, but already looking at this ab initio annotations, focusing on gene ontology terms, we find some interesting patterns. So for Arabia, we find that more genes are associated with telomere maintenance are occur in the genomes when you have species for species with higher karyotypes. And what we try to do now is looking more into gene expansion and losses, and this is all ongoing research. So this is basically what we currently do now in the group. As you can see, I don't have a lot of answers. I have a lot of questions. And if you have answers, I'm looking forward to hearing them. So to conclude on this part, we overall find more rearrangements that actually don't manifest as karyotypic changes. So we have way more structural variation than we would ever have predicted. And for the 3D genome structure, we find both changes and conservative parts of it. And how this could relate to speciation, we don't know yet. And we still need to know what the needles actually are and how they may interact. Good, so much about chromosomes. Let's move on to secondary contact zones. For this, I have to quickly take you on a quick journey through time, no worries. So this is Switzerland. As you may know it, if not, this is Switzerland. As it looks like now, but of course, it didn't look like this 24,000 years ago. In fact, back then, most of the country was glaciated. That also means most of the extant biodiversity didn't exist in the country, but survived in one or multiple glacial refuges. And since the retreat of the glaciers, many species have recolonized the country and often come into secondary contacts. And of course, secondary contact zones are amazing when you want to study evolutionary processes, right? We have an original population, they split, they come together. What happens? So if they don't have any barriers, they would fuse together. But often they have some barriers. And we end up having hybrid zones. And of course, if you have selection against hybrids, through, for example, reinforcing selection, you may end up having two species eventually. That brings us back to barriers. And I talked before mostly about post-zygotic barriers, genetic incompatibilities, but of course there's additional barriers such as habitat isolation and gametic isolation that preclude gene flow, right? And when it comes to barriers, we have to be aware that the strength of the barriers and impact of reproductive isolation may actually vary among the different species. So if habitat isolation is important in one species pair, it may be not important in another species pair. Similarly, barriers can actually interact with each other and further increase the overall degree of reproductive isolation. So if you only have habitat isolation, you may reduce gene flow to some degree. When adding mate choice to it, then this may potentially reduce gene flow much more. And this is what we try to understand in our systems. What are the barriers and how they interact? And talking, of course, about speciation as a process, we can think of it as a continuum. So starting with an early stage, where you have little reproductive isolation, the late stage, where you have complete reproductive isolation. Similarly for gene flow, beginning at a lot of gene flow, hybrids, etc. At the end, we should hopefully have something like this, where you have totally isolated species. And along this continuum, we can also imagine that, that we have increased phenotypic, genetic, ecological differentiation, which could help us to identify potential barriers. And especially this so-called late stage of speciation is still the gray zone for speciation research, because we don't often know what happens. How do we actually end up to the final product, different species, if at all? And what we do is we collect now different species pairs or subspecies pairs as well, and start to place them along this continuum. And we have tried, hopefully convince you that we have this case here to the right that falls within this gray zone of speciation. And we'll actually focus on the two systems here to the right. And we start with this one. This is the Revia-Aurealis system. Shown here are different subspecies that we have in the Alps in different colors, but also the Jura in Switzerland. In the Alps, in Switzerland, we have two subspecies, that's Adita and Isarica. And in the field, you could actually distinguish them by the presence and the absence of a small white dot in the black dot, which is, however, not always there. It's always a joy working on Revia. They have a 2-year life cycle. And this, I want to remind you, this all started as a master thesis. And now I have a PhD student, Patricia, continuing on it as a PhD. And initially, there were 2 contact zones described back in the heart of Switzerland here. And they were initially described by someone who collected there in the late 70s, early 80s. And he described in the sense, he was walking along the hiking path. He went from one subspecies to another with a lot of hybrids in between. And me and my master student were just curious, like, let's have a look 40 years later, what's going on there. We went to these 2 contact zones, one in the west, one in the east. We also collect allopatric sites. So in the north, we have Isarica and south, we have Adita. And what we can see is they differ in the way how they geographically isolated. The west is basically spatially connected, when in the east, the allopatric sites are separated by valleys. And this is what it looks like in the field. So imagine you stand here with this beautiful meadow full of butterflies in the allopatric side. You see on the other side of Lake Lucerne, this is the contact zone in the east. The other contact zone is there. To the right. And what you can also see is they really like very steep mountain slopes, which is a lot of fun to collect them. But we did that now since several years. And we look at different things. So trying to identify different barriers. So we look at male genital morphology. Here's just from the allopatric individuals. And what we can see, the 2 subspecies differ to some degree, but there's a lot of overlap. So, given that we have this strong phenotypic overlap, it's likely not a strong barrier. So the idea in butterfly is that they have this key lock mechanism, and therefore this could also be linked to mate choice. Adding some genomics, so now we have 350 resequenced individuals. This is the PC plot. We can see PC1 separates the 2 subspecies. We see that in the east, the spatially isolated one, we have 2 beautiful clusters. When in the west, that is the connected contact zone, we have basically a gradient. So that's fast. Sorry about that. So we have reduced gene flow in a spatially more isolated contact zone, suggesting that geographic isolation could be a barrier here. What about pheromones? So we looked at particular hydrocarbons, just summarize it here in a PCA. We see basically there's a lot of overlap. There's actually not much difference. And in a nutshell, we think that also there's the mate choice being as a weak barrier. So what could it be? What I haven't shown you yet are the contact zones. So back then, in 2018, when we went out and looked at morphology, we only got individuals that looked like this is Isarica subspecies. And we were like, contact zone is gone. But then we decided to go back for whatever reason the year after, and we saw something even weirder. We saw a phenotypic shift for something that looks like Isarica, something that looks like Adita. And the other contact zone, we found actually both phenotypes. And now we have years and years of collection, and we see these phenotypic shifts between even and odd years. But what happened, right? Because this is what we were supposed to find. This was what was described by this old entomologist, who luckily deposited his samples in the Natural History Museum. And we went back to the Natural History Museum and realized he didn't collect them just in 1 year, but over many years. And looking at the morphology here for wing morphology, we find the very same patterns. So we switch between something that looks like one subspecies in even years to another subspecies in odd years. To convince you more, we can also add now some genomic data. So we have the two contact zones with the allopatric left and right. What we see is that in even years, we have everything is blue, so it's Isarica. In odd years, we have also orange, so Adita, especially in the east, when we have hybrids and pure Adita also in the west, together with Isarica. So what we have here is a case of so-called allochronic isolation during secondary contact. So temporal isolation as a barrier to gene flow. And this is a case of asynchronous phenology, which seems to maintain secondary contact since at least 4 decades. We don't have the genomic data yet for the historical samples. This is work in progress, but yes, it's when you think you stand at the very same point from one year to another, and you just have another subspecies flying in your net. So the question was, did this evolve at the zone of secondary contact or not? To answer that, we went to the data of the Swiss Faunistic Institute. After cleaning, we had about 15,000 observational data points. Mind, they don't record subspecies. So what we did is we aggregated the data in 5 kilometer grids and just put a color on if they fly in even and odd years. And what you should see is basically we have a part of Switzerland that's primarily blue, another part that's primarily red. And this corresponds pretty well to the different subspecies. Now we actually wonder with with my PhD student, is this only something that happens in Switzerland? We went to get GBIF data, and sure enough, it's not the case. It's actually more widespread than we would have thought. So you basically see a map of Europe with patches of primarily blue to primarily red. So we have cases of this allochronic differentiation also, for example, here in France, in the Massif Central, between north and south, along an east-west gradient in the Pyrenees. Same for the Czech Republic, where they have 2 subspecies that actually have been described to differ in their phenology. So we try now to sequence individuals from all the different subspecies and look in a phylogenetic context to which degree this could have potentially evolved along this cryptic species complex. And you may have wondered, what were the chances that we find this? At least I did. So we went to the citizen science data of Infofauna for all the Swiss butterflies. They had more than 1 million observations. And guess what? There's only one other case, which is Aeneis glacialis. We haven't studied that yet, but yeah, that was really a lucky find. What about other barriers, right? So we actually assembled chromosome level genomes for both subspecies. And you see in the synteny plot, there's some rearrangements, but not that many. So you may think like, okay, it probably doesn't have an impact. But actually, Adita lost a W chromosome. And when we saw that, the first thing we did is we went back to the -80 to check if we really sequence a female. And I can assure it's a female. We took our resequence data, aligned it, and so this is for allopatric sites. We see that the allopatric Isarica, the females all have an increased coverage, so they all have the W chromosome, when for the Adita, they don't have the W chromosome. So this seems to be a larger pattern, at least in this part of Switzerland. We don't know yet what it looks like in the large geographic range, which is also something we get data soon. Going back to this plot, adding some colors, we actually also see that in the eastern contact zone, there's almost no gene flow, except for 2 F1 hybrids. But in the other contact zone, there's a gap. suggesting that backcrosses occur primarily from hybrids towards this aurica, but not the other way around. So this is all new. That's as far as we are. Nevertheless, we think that the loss of a W seems to be linked with asymmetric gene flow, so we may have sex-linked genetic incompatibilities. Those are the barriers that we so far investigated, putting this on a scale, what we think could be act as a barrier, what is not. We have some identified, some we still have to investigate. Now the question is, of course, what does it look like in other systems? I give you an example for the Tindarus system. So this is also part of the PhD of Hannah. So we have two closely related rabia species, that's Cassioides and Tindarus. Classically, in the 1950s, people did successfully F1 crosses, but didn't go further. Hybrids have been described based on phenotypes, but are very rare. And what we did is we also went out, collected across the distributions. So we had about 350 individuals, whole genome resequence data, and included 2 contact zones. And just to give you an idea what those contact zones look like, they're actually very narrow. So this is about 10 kilometers, and the zone where they fly together is just about 500 meters. That's it. And this zone is stable for at least since at least 1960s. That's the first time people looked into it. Nevertheless, the 2 species cannot coexist, it seems. Adding now the genomic data, we see also genetically they're very strongly differentiated. Actually, we only found 2 F1 hybrids in one of the contact zone, and the other contact zone, probably by chance, we didn't pick one up. Average genome-wide FSD is 0.7. It's not very surprising for butterflies. So we have strong interspecific differentiation of just a few F1 hybrids. And we also went through trying to identify different barriers, and we'll give you the summary afterwards. I just want to highlight one of them that we kind of were surprised to find. Because what we realized is when we sequenced the butterflies, we didn't just sequence the butterflies, but also some of their parasites, which in this case is Wolbachia. So Wolbachia has been suggested to be a driver of reductive isolation that can lead, for example, to cytoplasmatic incompatibilities, but we don't know yet what they do in our system. But why I'm talking about Wolbachia? So you will see now the structure analysis, mixture analysis for Wolbachia. And what we see is that all the Cassioides have Wolbachia. 2 F1 hybrids have Wolbachia, actually the strain from Cassioides. Alpine Tindarus, they have a high prevalence for Wolbachia, but at the contact zone, they have either lost it or reduced the prevalence significantly. So we have to acknowledge a very rare case of stable but asymmetric Wolbachia infection upon secondary contact. So to give you an idea what we looked into, so we have a lot of rearrangements. So they have actually the same karyotype, but it's this crazy example of many rearrangements that we see, which could be linked to incompatibilities. We have strong phenotypic differentiation, for example, male genitalia. So it could be linked to mate choice. They actually have strongly differentiated particular hydrocarbons, so they could recognize potentially each other. Nevertheless, they generate at times F1 hybrids. And they have a diversion Wolbachia infection. What they don't have is ecological differentiation. So all the ecological data that we looked at suggests they have pretty much the same ecological niche, which is likely the reason why they cannot coexist. And indeed, a recent phylogenetic analysis suggests that closely related rabia species have similar ecological niches more often than you would expect by chance. So we have a case of niche conservatism. Getting there. So to conclude on this part, we actually find various outcomes of secondary contacts. We have cases of hybrid swarms. I hadn't taken the time to show you this, but we have this Lycena hypotoa subspecies, and we actually found a hybrid swarm with capturing of one of the Z chromosomes. Two cases where we have totally isolated species that, however, cannot coexist. And we keep on adding different systems. So this year we collected 2 master students, individuals for 2 cryptic species in Switzerland, Ophidia saurinia and Lycina titirus, and which will get the genomic data soon. So I don't know yet what's going on there. And with that, we're almost at the end. Gives me time to give you the take-home messages in memes. So I hope I convinced you that holocentric chromosomes are actually interesting. And if you haven't considered about working on them, there's still time. Speciation genomics has now a 3-dimensional aspect to it. We don't yet know a lot what it can do, but there's now so much data around. Everyone can easily generate high-C data, and it's just, yeah, the peak of the iceberg. And when it comes to secondary contact zones in Arabia, they do weird things. They're totally not what you expect. They often don't hybridize, and which, at least to my understanding, contrasts what you would expect in other contact zones. And with that, there's many people that I need to thank, including people that fund me, and of course, you for your attention.
Yes, anyone have any questions?
Yes.
So do you know how recognition is working in these holocentric chromosomes? Like, is it?
It's a good question. Well, and it probably also depends. The main problem is we don't, so for Lepidoptera, we don't even know what the whole centromeres look like. So for in plants, the whole centric plants, we know, and then the whole centromeres also, differently spread across chromosomes, which can affect the recombination rate. So we have a project where we try to link 3D genome structure and chromosomal arrangements with recombination, but we don't have data. We have in the data generation phase.
Yeah, maybe it's a related question, but about the genetic architecture of this fusion and fission. Do you know if the species have different number of centromeres, and if they are in different places in the genome, and would it explain the difference in fusion and fission between the species?
It's actually one of the questions I was asked another professor working on holocentric chromosomes in plants, and he suggested yes, in plants they're not equally distributed, and if you have like a gap in the genome, it's probably more likely to be torn apart during cell division. But in Lepidoptera, we don't know it. It's literally, we don't know what the holocentromeres look like, but it's one of the guesses that I have as well.
I had a question, but this one might just speak to my lack of knowledge about like sexual reproduction in sex chromosomes. But so when they lose the W, does that not cause, like, I don't understand how then they even reproduce with each other. Like, how does that work?
I don't know yet. I mean, it's an interesting question, but what I haven't plotted it yet, or my PhD hasn't plotted yet. It's also if there's a sex dimorphism in terms of like hybridization, but we also find that not a lot of interaction, but we also find from the plot so far, it looks like we only have F1s, back crosses, and so on. So there seems to be there's some segregation issues associated, but this is all new, so this is all fresh, not even from the press.
Any questions on Zoom?
Not yet. I have a question. It's about chromo SSE. We went very fast on this method, and I did not well understood how it can differentiate between genesis and association?
But my PhD student really dig into the maps. So, but it's a SSE model that also estimates extinctions. So what I didn't tell you is also the groups that have the highest rate of diversification also have the highest rates of extinction. But then estimating extinction is also a bit debated to which degree the models work, but need to look at this. Hannah is the expert here.
Maybe related to the last question, I was wondering whether if in your system where there is a lost W chromosome, if there were a new sex chromosome system emerging, would you see it with the analysis you did already? Or?
I mean, so if you would have a new W chromosome, we would need to sequence more individuals with a chromosome level to actually assemble. I mean, assembling W chromosomes is not easy. So if there is a new W chromosome which would occur within the population, we would probably not see it because nothing would align against our assembly. For that, we will need to go for pack biosequencing, assemblies for large amount of individuals. It's possible.
Just studying.
Okay, well, I think you're off the hook, but yeah, if anyone wants to email me,
If you have answers or have some ideas what meals we miss, please let me know.
Thank you so much.
I was pretty bad at this question 2 years ago when I had more part ready.
Mais quand il s'est déconnecté.
Ah oui, c'est